This experimental system offers a high-throughput methodology for monitoring T-cell-mediated cytotoxicity at the single-cell level. First, label the effector T cells and tumor target cells with fluorescent dyes. Load both cell types onto the nanowell arrays and immerse the entire chip in medium containing Cytox green and fluorescently labeled annexin 5 in order to identify both necrotic and apoptotic cells. Now record an initial pre-image to determine well occupancy and cell viability at the start of the assay. After a 6-hour incubation, record a second image. Then use automated image analysis to quantify the antigen-specific lysis of EL4CD19 positive by CAR-positive T cells. Results obtained from this high-throughput method can screen antigen-specific lysis of targets by effector cells based on positive staining with Cytox and annexin 5.
The main advantage of this technique over existing methods like chromium-51 release assay is that true one-to-one effector-target interactions can be achieved and their cytolytic frequency measured.
The implication of this technique extends toward therapy of cancer because it allows to study correlates of efficacy in autologous cell therapy.
To fabricate nanowells using the silicon master, first thoroughly mix the Silgard 184 elastomer kit base and curing agent at a 10:1 weight ratio in a disposable cup using a plastic knife. Degas the mixture in a vacuum chamber for 1 hour, then pour the mixture onto the silicon master and let it sit for 30 minutes. Now, to cure the elastomer, heat the assembly in an 80 degrees